Any other, things that down the road or any other devices that either of you can talk about that you've got your hands on about vessel prep?
I can say that, a recent acquisition by Boston with Bolt, I mean, that's a laser IVL technology. They're going to have, hopefully long links. And so that'll be another IVL tool that's going to be out there as well for both coronary and peripheral applications. There's like 8 IVL companies out there, all who have different mousetraps as well too.
After the $13 billion acquisition there.
Exactly.
It's a nice space to be in.
Well, it's just kind of like the thrombectomy world. People just are now, this is their next thing, and there'll be something else. But BTK is like the Wild West, and everyone knows we're doing a bad job, but we need to do better. So I think I'm excited that they're investing more in this space. And, the long link resorbable scaffolds, I mean, R3 is going to be very interesting to me, I think, to see if that works, if that's successful. And also I'm interested, in potential newer applications of serration angioplasty, especially with drug, retrievable stent technology with drug. I mean, these are all things that are potentially coming. We just have to make a value argument for it.
I remember, is that, I think Fatty would show sometimes online, like, I think it must be the Recor where you can inject drug through the spikes or something.
No, that's a bullfrog device. No, that's
bullfrog. What is that?
Mercator, yeah. So they pivoted. Now they're actually in the venous space. So that was a balloon.
They're in the arterial space too. They're actively conducting another tibial trial.
Yeah, another trial. Yeah. They've been at it for a long time. They had a single spike that you could inject a physician-specified therapeutics. Usually, at this point, it was dexamethasone. When they first started, they were doing things like paclitaxel. But yeah, in the arterial space, they've been at it for a while. But the venous stuff, they're in the Dexterity AFB trials. They're looking at reducing rethrombosis post-venous thrombectomy and DVT to see if that might actually help, because rethrombosis is a major issue in that space. But the spikes that are on the spur are not hollow, they're solid. So in the drug version, the balloon will be coated and the spikes will be coated, and then those will be infused or injected into the medial adventitial space. The idea of bullfrog and 102 is still very interesting. I think the challenge becomes like, what drug do you use? And I think, Mike can correct me if I'm wrong, I think they're still using steroids.
The trial they're doing right now is a limus-based.
It is limus-based. Okay, good. Yeah. Good. That's what we had asked for years ago. So it's like, because they were having us try to use paclitaxel back in the day. So, but that's cool. I mean, that if you can show that it distributes evenly in that medial periadventitial space as well too, that might work really well. Then you just have to get it to stay there. Mike, do they have like a nanoparticle or anything like that to keep it around? Or, how do you prevent it from infusing away?
I think it's just because you're injecting it, when you inject it correctly, you're injecting it inside the vessel wall, you're sequentially injecting it along the length of your treatment area, right? So you're not necessarily injecting it in a place where it can necessarily move if done correctly. Obviously, if there's too much calcium and the needle can't penetrate into the wall, you're going to see that contrast and drug go downstream, right? Which is, ineffective.
Yeah, because they mix the contrast with the drug, right? When they're injecting.
They have like essentially a pneumatic pump that you use to put small amounts of the drug into the wall.
The big challenge with limus, as we're talking about, it's got a broad therapeutic window, but the problem is getting it to stay where you need it to stay and not just diffuse away into the reticular endothelial system, right? So the thing is, it goes to the vasa vasorum, then it disappears, right? So you want it to kind of hang around. So you typically need some kind of particle or something you encapsulate the limus in so that it hangs around and doesn't just disappear. Paclitaxel you could do that with because it's cytotoxic. It's directly on contact. So if you inject paclitaxel, it just kills things immediately. But with limus, it's a slow effect over time. So I'm curious how they did it or how that's going to go.
I mean, it's fun to see all this innovation and everything. I mean, we touched a little bit about how, this all adds time to a procedure. You do all of these interventions to the tibial and then adding all this. I mean, we're going to have to kind of figure out as we go on how to kind of optimize. I mean, adding so much more time to a case can be kind of hard if you have a busy day too, but the outcomes definitely outweigh, some of that. But it can be hard in a busy practice and whatnot. But yeah, I mean, that was a lot of really great info, guys. Any kind of last words of wisdom or people who are, maybe companies that are coming out with some devices, any kind of words of advice to our audience?
Well, it's not necessarily advice, but take this opportunity to to build on what you just said, it's the data that we have to generate that's going to help propel this field forward. It's going to justify using these specialty technologies. And, we haven't said it yet, but Jay and I are co-PIs for a prospective registry looking at serration angioplasty, the point force registry. Things like this, whether it's serration angioplasty or any of the other technologies that are coming down the line that Jay talked about, like these are the things that we have to do because we have to be physician scientists. We have to do the research. We have to be dedicated to changing the way therapies are delivered and which therapies are delivered. And I think that this is a great opportunity to talk about the work that we're doing, Jay, and, super proud to be doing it with you, someone I admire, with a company that I think is doing great things. Oh, man. But yeah. I'm looking forward to getting the patients we need in this trial and seeing how everything sort of shakes out.
And stuff like this is really exciting as well, too. So I encourage everyone to kind of listen in on these kind of podcasts. Honestly, I started doing BTK serration angioplasty because of Mike. So I saw his cases, especially below the ankle as well, too, because I think he had some of the early experiences with it, too. And I was like very skeptical. I was like, hey, can I get it there? And, I tried it out. I was like, hey, this actually works really well. Kudos to you and the great work you're doing. I would just tell folks kind of as a parting comment that, balloon angioplasty alone is not enough. I mean, we got to do better. And we know that like a lot of times we're undersizing our treatments, the vessel recoils ends up looking just the same as when we started or worse. So we got to find ways to do better for our CLTI patients. And, I look forward to these kind of technologies to helping us do that.